With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
In this question, we are asked that which of the following bacteria is not a source of restriction endonuclease. So restriction endonuclease is simply an enzyme, right? It's an enzyme that's also known as the molecular scissors, right? That's also known as the molecular scissors. So this enzyme or these molecular scissors, they are being used to cut the DNA, right? So as to cut the DNA at specific location. Right, so whenever we want a fragment of DNA, so we cut that fragment of DNA using these molecular scissors at the specific location. So this enzyme is simply used in cutting the DNA at specific location. So that's known as the restriction endonuclease. Now for this enzyme, so this enzyme is produced from different bacterial sources, right? So the restriction enzyme, that's E. coli. Right, the restriction enzyme, one of the example is E. coli, is isolated from the E. coli RY13, right? And the first restriction enzyme was the Hind2, right? The first one was Hind2. This was the first one, right? And it was simply isolated from the bacterium Haemophilus influenzae. Right, Haemophilus influenzas. So it was this enzyme. So this first enzyme is isolated from the Haemophilus influenzas. Right. Apart from this, if we discuss further, then the restriction enzyme BAM-HI. Right. This has been isolated from the Bacillus amyloli. Right. So if we look at the option here, so the option 1, that's Haemophilus influenzae, it's being the first source from which the first restriction endonuclease is isolated. That's the Hind2, correct. E. coli, from the E. coli, the restriction enzyme produced was E. coli. Right, and then from the we talk about the Ectobacterium tumefaciens. So it's basically a pathogen, right? It's a pathogen of several dicot plant. It delivers a piece of DNA known as tDNA in the TI plasmid, which transform normal plant cell to tumor cells to produce chemicals against the pathogen, right? So this was never a source of restriction endonuclease and will be a correct answer here. And as you've talked about the Bacillus amyloli crucifaciens, right? So it has produced the enzyme BAM-H1. Right, so simply we can say that it is the option 3. That's our correct answer here, right?
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.